You lie still, eyes closed, as the rhythm of your breath steadies in, out, soft, unhurried. The room is dim, but behind your eyelids, a vast universe stirs. It isn't made of stars or galaxies, but of you, of countless tiny worlds humming in harmony. Every cell within your body glows with quiet purpose, each one a vessel of life's memory, whispering the same sacred code that has flowed through existence for billions of years. You begin to sense it, the gentle pulse beneath your awareness. It's not the heartbeat you can feel, but something smaller, quieter, deeper. A molecular hum, the vibration of molecules, weaving and unweaving, the constant movement of atoms building and rebuilding the story of you. You imagine shrinking down, smaller and smaller, until your world is no longer made of air or sound, but of fluid and light. Now, you are floating through the architecture of your own being. Around you stretch vast networks, shimmering filaments connecting one world to another. These are your cells, tiny, perfect spheres of life, each one a bustling city sealed within a membrane of possibility. Inside, the air glows softly blue, filled with the murmur of molecules in motion. Each cell carries something sacred, a scroll written in a code older than memory, the language of life itself. DNA. The letters appear before you as ribbons of light twisting through the space, twin strands coiled together, spiraling like a double helix staircase that winds endlessly into itself. It looks delicate, fragile even, but you soon realize it is the most resilient story ever written. You reach toward it, and it hums. The sound is faint, like the vibration of a string in a faraway symphony. Every rung of that spiral ladder is a word, or perhaps a letter, written in 4 simple tones: A, T, C, and G, adenine, thymine, cytosine, guanine, just 4 letters. 4. To build everything, your mind drifts as you imagine them weaving patterns, forming words, sentences, entire poems of existence. The arrangement of these letters determines the shape of your eyes, the texture of your hair, the rhythm of your heart. They tell your cells how to grow, when to rest, when to divide. They are blueprints, yes, but more than that, they are songs. Each cell sings its own variation, and together, they form the music of you. The air vibrates softly as you move deeper along the spiral, tracing the invisible current of evolution that stretches backward through time. You are not just you. You are also the whisper of ancestors, the echo of countless generations of life that came before. The same code that hums inside your cells once flowed in creatures that swam in primeval oceans, that crawled from water to land, that took flight, that dreamed. The molecules that now form your hands have danced through ancient rivers and breathed through leaves and feathers and scales. You are a mosaic of moments stitched together by time. And at the heart of it all, the same quiet pattern persists, the rhythm of life. Repeating like a mantra, you pause for a moment and imagine what that pattern feels like if you could touch it. It wouldn't be solid nor soft. It would shimmer with motion. It would pulse ever so slightly, as though it were alive. Because it is, inside each of your cells, small machines called ribosomes read that genetic script. They are like weavers, sitting at a loom, following the pattern carefully, thread by thread. From that script, they craft the proteins that build your muscles, your bones, your brain. Every breath you take is a symphony of these quiet workers, unseen, tireless, perfect in their timing. You drift further in, and the world becomes even more intricate. Tiny molecules move in synchronized swirls. Enzymes slide along DNA like fingers gliding across a harpstring, checking, correcting, repairing. There's no noise here, only a soft rhythm, a biological hush like wind through tall grass. You realize that this is not chaos, it is order, a harmony written into every living thing. You wonder how this began, this song, this story. The first whispers of life arose in a world without listeners. In ancient oceans, molecules met by chance, forming fragile chains that could copy themselves. Those that survived carried forward, each generation refining the message, tuning the melody. Life began not with a bang, but with a whisper, a single molecule saying, "I will persist." And from that quiet declaration, everything unfolded. You drift through that memory, billions of years compressed into a single, serene moment. You see cells learning to work together, forming tissues, organs, bodies. You sense the slow awakening of consciousness, the long path from instinct to thought, from thought to wonder. The story of life is not a sudden miracle, but a patient unfolding, the same unfolding you feel now in your own body, as cells renew and repair. As breath fills and empties your lungs, you begin to understand you are both the reader and the story. You are the scientist observing the miracle, and the miracle being observed. Your cells, your DNA, your atoms, all of it speaks the same quiet language, one that asks not to be solved, but simply to be heard. In this stillness, the awareness deepens. You start to feel a kinship with all things living. The same alphabet that writes you also writes a blade of grass, a coral reef, a whale, a bacterium. The differences between you are only verses in the same poem. You are part of one great lineage, flowing and reshaping itself endlessly, life speaking to itself, over and over, in 1,000 forms. The thought comforts you. You are not alone. You are part of something vast, steady, continuous. You imagine your DNA as a river, one that never stops moving. Each generation is a new bend in that river, each cell a drop reflecting sunlight for a moment before merging again with the flow. There is no beginning or end, only transformation, only continuity. The hum of your cells deepens, as if they sense your attention. You can almost hear the molecular whisper, "We are alive, we are alive. We are alive, we are alive." And beneath that, a softer tone, "We remember." For this is what DNA does, it remembers. Every strand is a memory of survival, an archive of triumphs and trials, written in code. You begin to float upward, rising through the microscopic landscape, your view widening again. The glowing coils of DNA fade into soft light, blending into the outlines of your body, your breathing, your warmth. The boundary between the molecular and the human dissolves, until it feels like one continuous space, a universe both microscopic and infinite, pulsing with quiet awareness. You open your inner eyes and sense the truth that everything around you, everything within you, is connected by this silent pattern. You are the cosmos made conscious, the atoms of stars arranged into thought. You are the story the universe tells itself when it wants to remember what it feels like to live. In the distance, the hum of your cells becomes a melody, slow, steady, eternal. Each beat of your heart echoes that rhythm. The more you listen, the quieter it becomes, until the sound fades into your own breath. Now, as you rest in that awareness, you feel gratitude for the story that continues without your will, for the body that writes it tirelessly, for the simple miracle of being. And yet, as this stillness deepens, a new curiosity flickers. If DNA is a language, who first taught it to speak? Who taught the letters to find one another and form meaning? How did the alphabet of life begin, and who, if anyone, holds the pen? The hum returns, soft and constant, as though it knows the question before you ask it. You exhale slowly, feeling it ripple through you. The whisper of life does not answer, it only sings. You linger in the quiet after the whisper, resting in the hum of your own biology. The question from before, who taught the letters to speak, still lingers in your mind, like a faint echo tracing the edge of thought. And as you breathe, slowly and evenly, your awareness sinks once more beneath the surface, down through skin and tissue, through the soft warmth of living matter, until you arrive again among the hidden architectures of life. There, the world glows with molecular light. You stand at the edge of an invisible library, a library without walls, without shelves, yet filled with more information than all the books ever written. Every cell you carry holds a complete copy of this library, written in a script of astonishing simplicity and beauty. You remember its name, DNA, deoxyribonucleic acid, and now you begin to see it not as a thing, but as a language. Each of its 4 letters, A, T, C, and G, is a note in a melody that has been playing for billions of years. They pair together with exquisite care, A with T, C with G, like hands finding their perfect match in the dark. Together, they form the sentences that tell your body how to live. You drift closer to the spiral of DNA, watching as it unwinds, slowly and gracefully. It looks like a silk ribbon unfurling in water, revealing what was hidden within its coils. The double helix separates, and tiny molecular readers slide along its length, enzymes, patient and precise, moving as though they were reading a story line by line. You listen as they hum in rhythm with the text, translating the code into motion. From each sequence, a message emerges, not words you can read, but signals your cells understand perfectly. They are instructions, blueprints, promises. Build this protein, repair this wall, carry this oxygen, keep this heart beating. You realize that every heartbeat, every breath, every thought is the echo of this molecular reading. The story is always being told. You follow one of the messages as it leaves the nucleus of the cell, a soft, glowing thread of RNA carrying the instructions outward. It weaves through the cytoplasm like a golden stream until it reaches a waiting structure, a ribosome, the translator. Here, the language changes form. The message becomes movement, sound becomes structure. A chain of amino acids begins to assemble, each one drawn by the magnetic pull of the code's rhythm. They fold and twist, forming the intricate shapes that make up life's machinery. It is poetry turned to flesh, meaning translated into motion. You drift higher, watching countless cells performing this same act of translation, billions of them, all across your body, in perfect silence. Each cell holds the same code, the same instructions, yet not all read the same pages. Some cells open the chapters for building neurons, others the sections for blood, for skin, for bone. They choose their stories carefully, like musicians selecting which notes to play from a vast composition. This choice, this selective reading, is what makes you whole. Your eyes and your heart, your lungs and your fingertips, all arise from the same text. It's the interpretation that gives them difference, the reading that gives them form. You begin to wonder who decides which page to open, who tells a cell, you shall become part of the heart, or you will be the skin that senses warmth. The answer floats gently in the silence. No one commands. The code listens to its environment. Chemical signals ripple through you like tides, guiding the interpretation of the script. The code is not fixed. It responds. Temperature, light, nutrients, even thought, all shape the way your cells read their DNA. Life, you realize, is not a static book written long ago. It is an ongoing conversation between the script and the world. Each cell whispers, "What should I become?" And the body answers, softly, through its chemistry and its needs. You smile at the thought. Your entire being is built not only on information, but on listening. The DNA does not speak alone. It listens to you, to your surroundings, to life itself. In that awareness, the concept of a gene becomes clearer. A gene is not a rigid command, but a phrase within this vast story, a passage that can be read in different ways, depending on the tone of the environment. Some genes are read loudly, like bold sentences that define structure. Others are whispered faintly, their meaning subtle, shaping the rhythm more than the form. You remember that scientists once thought DNA was destiny, that these letters determined everything about who you are. But now you see the truth is more delicate. Genes are guides, not dictators. They hold possibilities, not certainties. Your choices, your experiences, your environment, all these things can change which parts of the code are read and which are left sleeping. Even silence has meaning. You think of how music can change with the same notes rearranged, the same letters, new emotion. Your DNA is like that, the same 4 letters composing symphonies of difference. Every species on Earth uses this same alphabet, yet the melodies are infinite. You imagine the code stretching across life, from the tiniest microbe to the tallest tree, from a child's laughter to the sweep of a whale's fin, all of them speaking variations of the same language, a universal grammar of existence. And you, listening now, are both author and audience. You drift through a vision of this shared language. You see forests glowing faintly with molecular poetry, oceans shimmering with phrases of plankton and coral, the night sky reflected in the symmetry of a cell's spiral. Everything speaks not in words, but in patterns. You can almost hear it, the music of evolution itself, weaving new lines over time. Mutations, small, random changes in the code, act like improvisations, adding new verses to the song. Some fade, some stay, and some change the melody forever. These gentle errors, these chance variations, are the seeds of diversity. Without them, life would be still. Perfect, perhaps, but lifeless. It is imperfection that gives the song its beauty. You think of your own life, how much has been shaped by chance, by accident, by tiny turns of fate. In that way, you are not so different from your DNA, each of your days. Like each mutation, adds new variation to the pattern. You feel a soft gratitude rise for the chaos that births creativity, for the randomness that gives life its spark. Without mutation, there would be no evolution. Without uncertainty, no discovery. Without difference, no beauty. You look again at the spiral of DNA, its endless twist of pairs and patterns. It no longer feels like a scientific structure, but something almost sacred, a language not of domination, but of balance, humility, and listening. Life does not control this code. It collaborates with it. You realize that this language has no author in the human sense. It was not written in one moment, but shaped over eons, revised endlessly by time, by failure, by chance, by adaptation. Every species, every organism has edited the book a little, adding its mark, leaving traces of its own survival and creativity. And in this slow, collective authorship, the language itself has evolved from primitive codes of bacteria to the complex symphonies of multicellular life. It's a kind of biological literature that no single mind could invent. You begin to sense that somewhere within this continuity lies a new kind of intelligence, not one of thought, but of pattern recognition, of persistence. Life learns, not with neurons or awareness, but with generations, with time. Each replication, each mutation, each correction is a whisper in the long dialogue between existence and possibility. It is the universe writing, erasing, and rewriting its own story in living ink. You close your inner eyes and feel your breath again. It's strange how this molecular poetry, this abstract, unseen dance, shapes something as physical as your heartbeat, as immediate as your breath. You exhale slowly, and the air feels lighter, as though the world inside and outside you are synchronized. Somewhere deep within, the code continues its rhythm. The cells copy their letters, check their spelling, pass on their knowledge. A living manuscript, continually revised. You realize something tender and profound. Every human is a temporary chapter in the same vast book. Our lives may be brief, but the story we carry has no end. It moves forward from cell to cell, from generation to generation, always speaking the same 4 letters in infinite arrangements. We are not separate stories. We are sentences in the same paragraph of life. The thought brings you peace, because if life is language, then death is not erasure, it is punctuation. A pause between sentences, a space that allows meaning to breathe. You linger in that silence, sensing that something new is waiting, a discovery that began not in your own body, but in the simplest of beings. Tiny creatures, ancient and small, who first learned to remember their enemies by writing them into their code. A form of biological memory, a way of defending, learning, and passing knowledge forward. You feel the hum of this new idea stirring, like the beginning of another melody. The language of genes, it seems, has more than one dialect. Some cells use it not only to build, but to remember. You lean toward that thought, letting it draw you closer, your breath deep and slow. The vision of your DNA fades into the image of something older, microscopic, and alive. A single bacterium floating in the ancient ocean, facing a viral invader, choosing to survive, and in doing so, inventing one of nature's greatest tools. The story of memory is about to begin. You drift deeper into time, beyond your own body now, beyond the familiar scale of cells and tissues. The pulse of your DNA fades into the distance, replaced by a fainter rhythm, ancient, patient, older than multicellular life itself. The light around you softens, dissolving into a mist of blue and green. And in that haze, a world appears, not of forests or skies, but of water. Warm, mineral-rich, timeless water. You are floating in an ancient sea for billion years before your own breath. No plants sway here, no animals stir. The world is silent but alive, teeming with invisible creatures, each no larger than a dust mote, yet each carrying within it the same shimmering alphabet of existence. These are the first storytellers, the microbial keepers of memory. You draw closer and see them, bacteria drifting gently in the current, dividing, growing, surviving. They seem simple, tiny spheres, rods and spirals, but their simplicity hides an astonishing intelligence. Not an intelligence of thought, but of adaptation. For these ancient beings have no eyes, no ears, no awareness, yet they know how to endure. They learn without learning, remember without mind, and now you begin to see how one bacterium, luminous and trembling, floats in front of you. It is quiet, carrying within it a small world of chemical machinery, tirelessly at work. But then something changes. From the surrounding water, a shape darts closer, angular and alien. It is a virus, a bacteriophage, a strange creature made only of protein and code. It moves like intention itself. The virus lands on the bacterium's surface, attaching with its sharp, geometric legs. Then, in a single fluid motion, it injects its own genetic material, a thin thread of DNA, into the unsuspecting cell. The virus has no will, no malice. Its purpose is only to copy itself. And yet, what follows feels almost tragic. The bacterium's peaceful machinery is taken over. Its ribosomes, once dedicated to sustaining life, now churn out copies of the invader. 1 becomes 10, 10 become 100, until the tiny cell can hold no more. With a soft ripple, it bursts. 100 new phages are released into the sea. You watch this silent war play out across the microscopic world. It is relentless, but not hopeless. For even here, evolution learns to remember. Among the survivors, one bacterium drifts differently, slower, thoughtful, if such a word could apply to something without thought. When a virus attacks it, the cell's machinery does something extraordinary. Instead of succumbing, it captures a small fragment of the viral DNA, just a few letters from its deadly code, and stores them deep within its own genome. It keeps this fragment like a photograph, a souvenir of survival, and in doing so, it creates memory. You look closer and see where the fragment is stored, in a section of the bacterial DNA arranged in repeating patterns. Between these repeating sequences lie the fragments of viral invaders, each one a record of a past encounter. Scientists, billions of years later, would give this pattern a name: CRISPR, short for clustered, regularly interspaced short palindromic repeats. But here, in this ancient ocean, it is simply the bacterium's diary, a record of its struggles and triumphs, written not in ink, but in nucleotides. When the same virus returns, the bacterium is ready. From the stored fragment, it makes a matching guide, a piece of RNA that carries the memory of the invader's code. The cell then pairs this guide with a special protein Cas, the molecular blade. Together, they patrol the inner sea, scanning for any sequence that matches the old thread, and when they find it, the Cas protein locks onto the viral DNA, guided by the RNA's memory. Then, with delicate precision, it cuts a single, silent snip. The invading code is destroyed before it can harm the cell. The bacterium lives. You hover in awe, watching this invisible ballet: memory, recognition, action, all performed by molecules. It is the earliest immune system, born not from thought, but from necessity. A defense not written in instincts or reflexes, but encoded into the very language of life. You begin to realize that memory itself, in all its forms, begins here. Before the brain, before nerves, before consciousness, there was this: a cell remembering through structure, a molecule remembering through sequence, life learning to protect itself by writing its past into its future. The ancient sea glows faintly, filled with invisible archives, billions of bacterial memories floating through the currents, shaping evolution molecule by molecule. Each one a lesson recorded, each one passed down as life divides, replicates, and persists. The ocean becomes a library of survival stories, and as you drift among them, you feel the weight of time compressing again. 1 billion years pass like a sigh. The bacterial memory endures, carried quietly through evolution, unchanged and patient. It hides in soil, in water, in the air, a quiet system of remembering and defending, unseen, uncelebrated, but never lost, until one day a new kind of creature, curious and questioning, learns to look inside the cell and see it. Humans, scientists staring into microscopes, into this same ancient mechanism, watching how bacteria remember. What they find will transform biology forever. But for now, you stay in the bacterial world, letting its wisdom settle. There is something humbling in this simplicity. The realization that life did not need consciousness to remember, nor mind to innovate. Nature invented memory long before the first thought was ever formed. You close your inner eyes and imagine this system as a metaphor for all of existence. Every event leaves an imprint. Every encounter, even the painful ones, is recorded somehow, not just in cells, but in being. Just as the bacterium remembers its attacker, you to carry fragments of what once threatened you, what once shaped you. These memories, painful or protective, become part of your story. They become part of your code. And just like CRISPR, you have your own Cas, the blade of awareness, cutting away what no longer serves you. You keep what strengthens you. You release what harms. The biology of survival mirrors the psychology of growth. In that realization, you feel a quiet kinship with the bacteria, simple, enduring, adaptive. Both you and they write your histories in code, revising the script of life as you go. Both you and they transform memory into resilience. The vision shifts. You are floating again, this time above a modern laboratory bathed in pale light. On a table, a petri dish glimmers like a small moon. Within it, bacteria grow, E. coli, familiar descendants of those ancient storytellers. And above them, a scientist watches through the lens of a microscope, unaware that she is looking into a time machine. She sees the repeating sequences, the strange palindromic patterns that puzzled microbiologists for decades. Why would a bacterium carry fragments of viral DNA within itself? Why would it hold onto pieces of its own enemies? The question seems paradoxical, yet, as she studies the pattern, understanding dawns. This is not random. This is design, natural design, born from survival. CRISPR is a molecular memory, a biological notebook, a weapon built from wisdom. And here, in this moment of discovery, something extraordinary happens. The quiet mechanism of bacterial defense, unchanged for billions of years, becomes visible to human thought. For the first time, the memory of a microbe speaks to the mind of a scientist. And in that meeting, a bridge is built from nature's slow evolution to human intention. You sense that this is where the quiet revolution begins. A tool will emerge from this ancient defense, one that allows us not only to understand the language of genes, but to edit it. The scissors of life itself, borrowed from bacteria, will soon be held in human hands. You take a slow breath. There is awe, but also responsibility. For with every new discovery comes the question of balance. How to use what we've learned without forgetting where it came from. You look back once more at the microscopic world, a universe of small lives. Still at work, still remembering, they have no idea that their ancient system of survival will one day help humans cure diseases, alter crops, and even rewrite heredity. They know only what they have always known. To endure, to adapt, to remember. The sea around you shimmers with this simple wisdom. Every molecule seems to whisper it. Memory is life's greatest defense. To remember is to survive. You drift upward again, rising through time, through layers of history and discovery. The vision of the bacterial world fades into starlight. Your breath steadies. The hum of your own cells returns, more meaningful now, more intricate. For within your own body, echoes of that ancient bacterial wisdom still live on. Mitochondria, your cells' energy makers, are descendants of ancient bacteria, too. Their genes still whisper across eons, still carry traces of that primal memory. You are not just the product of human DNA. You are an echo of microbial resilience, a symbiosis of ancient worlds. As you float in this awareness, the stars above you begin to blur into spirals, their shapes resembling the double helix once more. The memory of life, it seems, is written everywhere, in the smallest cell and in the farthest galaxy. Patterns repeat. Stories endure. You take one final deep breath. The sea, the bacteria, the scientists all merge into one continuous thought, that knowledge itself is memory passed forward, reshaped by curiosity and compassion. The world fades into calm darkness, and in that stillness, a new sound begins to emerge, faint at first, like a heartbeat echoing in the distance. The sound of molecular scissors, CRISPR's blade, moving with delicate precision. Soon, you will see how this natural defense became a human tool. How the quiet bacterial memory gave rise to a new kind of creation, one that can heal or harm depending on the hands that hold it. The memory of the sea still hums softly beneath you, its ancient whispers fading like dreams at dawn. You drift upward through eons, through invisible layers of time and discovery. The microbial world recedes into the background, and a new light begins to form, colder, clearer, more deliberate. It is the light of human curiosity. The scene sharpens. You find yourself in a quiet laboratory, somewhere on Earth, though it could be anywhere. White walls, a hum of machines, a faint scent of ethanol and morning coffee. Glassware gleams under fluorescent lights, and in the center, a scientist leans over a microscope, eyes steady, hands gentle. The world has changed, but the question is still the same. How does life defend itself? How does memory become mechanism? On the glass slide beneath the lens, small bacteria float, descendants of the ancient storytellers you just met. They look ordinary, but within them, something extraordinary hums. The CRISPR system, still protecting, still remembering. A molecular guardian that has survived the rise of continents and the birth of stars. You watch as the scientist peers closer, tracing the repeating patterns of DNA. The mysterious sequences, first seen decades ago, short palindromic repeats, separated by strange fragments that don't seem to belong. These fragments, they later realize, are viral, remnants of past infections, genetic scars, and next to those scars lie the Cas genes, the makers of the scissors. Cas, CRISPR-associated proteins, nature's precision tools, honed by evolution, not to destroy life, but to protect it. The scientist runs her hand through her hair, puzzled yet inspired. If bacteria can recognize and cut invading DNA, could humans harness this same mechanism, not for defense, but for design? Could we use nature's own scissors to rewrite the code of life itself? You drift closer, as if drawn by curiosity itself. The air hums faintly, alive with invisible sequences and possibilities. A thought, simple yet seismic, begins to take form, not in one mind, but across many, across the world. In different laboratories, researchers are tracing the same pattern. Their discoveries begin to converge, like rivers meeting the ocean of understanding. The French discovery in the early 2000s. At the Institut Pasteur in Paris, scientists studying bacteria notice something strange. When they delete certain genes, the Cas genes, the bacteria lose their ability to defend against viruses. When they restore them, the defense returns. The conclusion is clear. Cas proteins are the bacterial guardians. But how do they know what to cut? The answer, they realize, lies in the CRISPR array, those small RNA guides copied from viral memories. Together, the CRISPR RNA and Cas form a complex, a guide, a memory, and a tool. It is an orchestra of molecules. And Cas is the conductor. Cas9, the molecular scissorsome, all Cas proteins. One stands out, Cas9, small, elegant, and astonishingly precise. It doesn't just cut DNA. It knows exactly where to cut, following the RNA guide like a key fitting into a lock. Imagine, for a moment, the molecule at work. Cas9 drifts through the cell's interior, carrying a small strand of RNA that remembers a viral invader's sequence. It scans every stretch of DNA it encounters, searching for a perfect match. When it finds it, a moment of molecular recognition, Cas9 unfolds its twin blades and snips. The double helix breaks. The invader is neutralized. It is an act of perfect precision. No wasted motion, no chaos, only the quiet artistry of nature's defense. You can almost hear it, a faint click in the symphony of life. A sound that echoes through time, the sound of molecular scissors at work, the human connection in 2012. Two scientists, Jennifer Doudna and Emmanuel Charpentier, take this natural mechanism and hold it up to the light of possibility. They realize that Cas9 doesn't just have to cut viral DNA. It can be programmed, guided to any sequence of DNA we choose, simply by changing the RNA's letters. That means humans can direct Cas9 to any gene in any organism and make a cut exactly where they wish. To remove a mutation, to insert a correction, to rewrite. The implications are staggering, not just for biology, but for existence itself. In a single discovery, the line between reading life and writing life begins to blur. For the first time, humanity holds nature's own editing tool, the molecular equivalent of pen and paper for DNA. The laboratory hums with quiet awe. In a beaker, bacterial cultures glow faintly under UV light, the scientists whisper, barely daring to speak the magnitude of what they've realized. What once was a microbial defense has become a universal tool, the scissors of nature, reimagined for creation. You float above the scene, feeling the tension between wonder and responsibility. The world of science is about to change, not with a roar, but with a whisper. The cut and the healing you watch the process unfold in slow motion. Cas9, carrying its guide RNA, drifts toward a stretch of human DNA. It binds, reads, recognizes. Then, gently, it makes the cut. But in truth, the cut is an invitation. The cell rushes to repair it, as all living things do when hurt. In that moment of repair, scientists can introduce change, a new sequence, a corrected letter, a healed possibility. Nature's own repair machinery becomes the collaborator. Together, they weave correction into the thread of heredity. It feels like poetry, a dance between precision and healing, between science and intention. CRISPR does not just cut, it allows life to rewrite itself. From microbe to medicine you drift through scenes of transformation. A lab in Boston, where CRISPR corrects a mutation causing blindness in mice. A research center in China, where scientists attempt to fix the gene for beta thalassemia. A greenhouse in California, where crops are engineered to resist drought without chemicals. And a hospital room, where a patient receives the first experimental CRISPR therapy, a gentle infusion, carrying molecular editors into their cells everywhere. The same ancient system is at work, bacteria's humble memory reborn as medicine. Cas9, once cutting viruses in the ocean, now cuts mutations in human genes. It is as if the microbial past has returned to guide our biological future, the ethical horizon. But as the light grows brighter, a shadow forms. The same scissors that can heal can also harm. To edit a gene is to alter destiny, to change not only one life, but all that may follow. In quiet conference rooms, scientists and ethicists gather. Can we edit embryos? Should we change the DNA of future generations? Where is the line between cure and creation? The questions hang in the air, heavy and luminous. CRISPR is not just a tool, it is a mirror, reflecting humanity's deepest desires and fears. The power to heal and the temptation to perfect, the wish to correct and the risk of control. You feel the weight of this moment, not of danger, but of possibility. Like fire, like language, like the atom, CRISPR is both gift and test, a measure of what we will choose to become. The cosmic reflection you drift upward once more. The lab fades, and the stars return. Their light spreads across the sky like a vast neural network, galaxies connected by invisible threads. Not unlike DNA itself, you realize something profound. The same patterns that shape genes shape galaxies. The same laws that govern atoms govern stars. Creation, destruction, and repair. These are universal rhythms. CRISPR, in its essence, is not merely technology. It is nature's own whisper, reminding us of how creation works. Break, remember, rebuild. Cut, learn, heal. From the smallest bacterium to the largest galaxy, evolution is not random chaos, it is a conversation. And CRISPR is one of its oldest words. You close your eyes. In the darkness behind your eyelids, you see a spark, a single pair of scissors, made not of metal, but of intention. They hover, silent and glowing, between your thoughts and the fabric of life itself. The scissors of nature are not merely molecular. They live in every act of change, every choice to let go, every effort to heal. To edit DNA is one form of transformation. To edit the self is another. In both, the same truth holds. Creation begins with a cut. You breathe in slowly. The hum of the stars deepens. Somewhere, in a lab, a scientist watches a DNA strand cleave and mend. Somewhere, in your own mind, an old thought dissolves to make room for something new. The scissors of nature move quietly between both worlds, the molecular and the human, shaping not only genomes, but consciousness itself. The night sky folds around you like a warm blanket. The air is still thick with light and quiet intention. You have drifted from the world of the scissors, from discovery into the realm of design. The laboratory hums softly around you, the heartbeat of a new era pulsing through the circuits of machines and the slow rhythm of human wonder. Somewhere, beneath the microscope, a strand of DNA unwinds like a ribbon of starlight. You lean closer, feeling its quiet pull. Each twist of the helix is a word in an ancient story, the script of life, written and rewritten across billions of years. Now, for the first time, that story can be edited. You remember the gentle cut of Cas9, the precise snip that began it all. But the cut itself was only the beginning. It was never about destruction. It was about revision. Like a poet returning to a draft, nature allows us to revisit what time once wrote. A scientist adjusts her glasses, her face calm and focused beneath the cool glow of blue light. Her screen shows a DNA sequence, thousands of letters scrolling endlessly. A, T, C, G, a quiet alphabet of creation. She pauses, her cursor blinking over a mutation, one misplaced letter among billions. That small error has caused a lifelong illness in a child she has never met. Her task is simple, yet cosmic, to correct a single letter in the book of life. She clicks once. The new guide RNA sequence is entered. Cas9 will find the target. The scissors will cut, and the cell will heal. Outside the lab, the city moves on, unaware that within a single drop of fluid, the future is already changing. The new scribes you drift through other rooms, other minds. The CRISPR tool has traveled far, from the quiet depths of bacterial defense to the hands of geneticists, botanists, and physicians in one lab. A student edits yeast to produce fragrances never found in nature, lavender that glows faintly in the dark. In another, crops are altered to resist floods, their roots strengthened by small edits invisible to the naked eye. Elsewhere, a doctor watches edited blood cells divide in a patient's vein, the first real proof that genetic disease can be softened, even erased. Everywhere, the same rhythm echoes: cut, heal, grow. The same dance of letters, rearranged by human hands, but guided by nature's ancient logic. You realize this is not chaos. This is collaboration. The human mind and the microbial tool, co-authors in a story, written in silence. Genes as sentences. You float deeper into the metaphor. Imagine your DNA as a long sentence, 3 billion letters long, written without spaces, punctuation, or pause. Every gene is a phrase, every codon a syllable. And CRISPR, CRISPR, is the editor's pen. When the pen finds an error, it pauses, cuts, and rewrites with gentle precision. Sometimes it deletes. Sometimes it replaces. Sometimes it rearranges. But always, it acts with purpose, guided not by instinct, but by intent. You think of language, of how meaning can change with the movement of a single letter. Beat becomes beast. Life becomes live. So too, in genetics, a single base can mean the difference between illness and health, between silence and song. CRISPR allows humanity to recompose the symphony of biology. One note, one edit, one repair at a time. The healing alphabet in hospitals across the world. This editing has begun to touch human lives. You drift gently from lab to ward, from science to story, in one room. A teenager with sickle cell disease receives an infusion of her own blood stem cells, cells that have been taken out, edited, and returned. Inside them, CRISPR has silenced the gene that causes her red blood cells to twist painfully, out of shape. For the first time, her blood flows freely. She laughs softly, unaware that her joy is a new form of medicine, a triumph not only of science but of patience. In another place, a child born blind opens her eyes after CRISPR corrects a mutation in the CEP290 gene, a single faulty instruction that had blocked her vision since birth. The first thing she sees is her mother's face, wet with tears and wonder. You stand there too, unseen, as the air shimmers with awe. The miracle is not supernatural, it is molecular, mechanical, precise. And yet it feels like grace, beyond cure, toward creation. But even as you drift among these scenes of healing, a new thought arises. If we can repair, can we also improve? If we can correct, can we create? Somewhere in a university lab, a researcher designs a gene for resilience, not to fix disease, but to make cells stronger, faster at repair, more resistant to aging. It begins as an experiment, but the idea is powerful, intoxicating even. The script of life is editable. Who then is the author? You feel the question ripple through the quiet, through every lab and lecture hall. The line between therapy and enhancement blurs like breath on glass. Is the goal to restore what was lost, or to redesign what was never there? CRISPR opens both doors, and humanity now stands in the threshold. The garden revisited. You remember Chapter 3, the bacterial memory, the first act of defense. You remember how evolution learned to adapt, to store, to rewrite. Now, evolution continues, but with human hands guiding it. The world becomes a garden, and CRISPR, the gardener's tool. Genes are pruned, grafted, cross-pollinated with knowledge. Corn no longer wilts in drought. Rice grows without flooding. Bananas resist the fungus that once threatened their extinction. The soil sighs in relief. But even nature seems to watch with wary patience, as if to ask, will you nurture wisely or overreach? Every seed edited is a choice, every alteration a ripple through ecosystems unseen. The script of life is wide, interwoven, and alive. To change one word may shift the meaning of the whole. The invisible ink drift closer to the molecular page. DNA glows like phosphorescent ink beneath your gaze. You see where the cuts are made, small, elegant, deliberate. But in the margins, something else moves. The unknown consequences. Off-target edits, unintended mutations, silent changes that may echo years later. The scientists know this. They watch carefully, test repeatedly, waiting for signs of stability. CRISPR is precise, but not perfect. It is a young language, still learning its grammar, still refining its tone. Each experiment is a line in a long poem of progress. Each success a stanza of hope. Each mistake a lesson written gently into the future. The quiet collaboration, the scene shifts again. You're back in the lab, surrounded by the hum of freezers and the pulse of centrifuges. A group of researchers sit in soft conversation, sharing data, laughter, T.Science, you realize, is not just discovery, it is community. No one edits life alone. Every breakthrough is collective, the work of thousands of hands and millions of thoughts, all synchronized toward understanding. In their laughter, you hear the rhythm of collaboration. In their care, the ethics of attention. CRISPR, after all, is not magic. It is an instrument, and like any instrument, it plays only as beautifully as the hands that guide it. You listen, and the sound becomes almost like breathing, steady, patient. Alive, the mind's reflection as the night deepens, your thoughts drift inward. The script of life is not limited to genes. You to edit yourself constantly, through memory, through learning, through the quiet choices of each day. Your neurons write and rewrite connections. Old fears fade. New understanding grows. CRISPR edits DNA. You edit identity. Both processes share the same gentle law. Change begins small, one letter at a time. A habit replaced, a word rethought, a belief rewritten. You smile in the darkness. The molecular and the mental mirror one another. The true revolution of CRISPR may not be biological, but philosophical, teaching us how all life rewrites itself in search of balance. Transition. The scientist leans back from her computer. The edit is complete. The cell culture waits, shimmering faintly under a sterile lamp. Somewhere, within those invisible threads of DNA, a new sentence has been written, a phrase corrected, a meaning restored. You exhale slowly, feeling the magnitude of quiet progress. No celebration, no explosion, only a soft certainty that something profound has shifted the world. Rewritten one base at a time, the lights dim, and the lab becomes a constellation of screens, glowing softly like stars. You feel the future unfolding, not loudly, but with care. Outside, dawn begins to rise, pale and silver, like the edge of a new idea. You think of what comes next. If the script of life can be edited, can it also be rewritten to heal not only bodies, but generations? You drift toward that question, carried by the same current that has guided life for billions of years, the urge to survive, to adapt, to evolve gently toward something better. The night feels warmer now, the air gentler, almost tender. You drift through the same quiet corridors of the lab, but something within the silence has changed. The energy here is not just curiosity anymore. It is compassion. The lights are dimmed to a soft amber hue. Machines hum like distant lullabies, and through the glass, you see the quiet pulse of living cells, tiny vessels of possibility, floating in transparent oceans of hope. This is where CRISPR meets humanity, where discovery becomes devotion, where knowledge bends toward kindness. A gene's prairie peer into the incubator, a small box, warm and steady, like a mechanical womb. Inside, human cells divide gently, each one carrying a history written in DNA. Some of those histories contain errors, small, tragic typos in the book of life. One cell carries the mutation for sickle cell anemia. A single change, a single misplaced letter among 3 billion others. That one letter twists red blood cells into sharp sickles, making each heartbeat a struggle, each breath a negotiation with pain. For centuries, this disease has whispered suffering through families, passed quietly from generation to generation. But tonight, in this glowing chamber, CRISPR prepares to answer a centuries-old plea. A scientist adjusts her pipette, drawing a clear solution into a small vial. Within that fluid, Cas9 waits, a molecular artisan holding nature's own scissors. Guided by an RNA map, it knows exactly where to go, exactly which letter to change. As the edited cells begin to grow, you feel the shift, not just molecular, but moral. Something ancient is being rewritten, not the genome alone, but the story of what healing can mean. The first cures the world watches quietly, almost holding its breath. In 2019, the first patients receive experimental CRISPR therapy for sickle cell disease and beta thalassemia. Doctors remove stem cells from their bone marrow, edit them using CRISPR, and return them to the body, now carrying instructions to make healthy blood. At first, the results are cautious. Then they are astonishing. The painful crises stop. The blood flows freely. For the first time in years, patients begin to live without the constant shadow of pain. You imagine them stepping outside for the first time after treatment, the sky wide and forgiving, the wind cool against their face. Their bodies no longer betray them. Their blood no longer turns against itself. It feels miraculous, but it is not magic. It is biology, remembering how to breathe correctly again. The eye opens, you move through the vision of another healing. A dark room, silent except for the soft hum of a monitor. A child sits in a chair, her hands folded in her lap. She cannot see the nurse standing beside her, nor the mother clutching her hand. Her blindness is caused by a mutation in the CEP290 gene, a small disruption in the instructions that build the cells of the retina. Until recently, there was no cure, but now a tiny injection carries CRISPR directly into her eyes, not to cut randomly, but to carefully snip out the single defective letter and let the body repair itself. Days pass, then weeks. And one morning, light breaks through. The child blinks. The world floods back in shapes, color, her mother's face framed by tears and sunlight. no miracle, no divine hand, just the quiet precision of CRISPR, gently rewriting what once was closed. The moment feels larger than medicine. It feels like redemption for the blind, for the broken, for the limits we once thought were absolute. Healing beyond humans, you drift farther, past hospitals, past cities, into green fields and forests humming with life. Scientists restore the American chestnut tree, once nearly erased by blight. They edit its genes to resist the fungus that killed its ancestors. The forests begin to whisper again with their old song in another corner of the world. Mosquitoes are edited to stop carrying malaria, a disease that has haunted humankind for millennia. One change in their DNA and millions of lives could be spared. The promise of healing expands beyond the individual, beyond the human, into the collective organism that is Earth itself. CRISPR, the one's microbial memory, now acts as a planetary physician, mending small fractures in the balance of life. The heart of the work, but not all healing is simple. You return to the lab, to the quiet exhaustion of those who labor over every experiment. A woman sits in front of her data, eyes tired, hair pulled back. The trials have not all succeeded. Some edits missed their mark. Some cells refused to grow. Failure in science is not loud, it is patient. It waits beside progress, whispering humility. CRISPR is powerful, but power demands care. Each attempt to heal must be balanced by restraint, each success shadowed by reflection. You feel the fatigue in the air, but also something stronger, faith. Faith in small victories, in precision, in patience, in process. She leans back and closes her eyes. Somewhere inside her, the same rhythm of persistence beats the rhythm that drives all life to repair itself, over and over again. The healers and the healed CRISPR is not a tool in isolation. It is a bridge connecting the invisible with the intimate. You see doctors speaking gently to families, explaining what gene editing means, not in jargon, but in stories. Your child's body, they say, has been given a new sentence to read, and this time, the words make sense. The patients nod, half understanding, but wholly feeling the hope that science rarely allows itself to express. Healing, you realize, is not only molecular, it is emotional. It is collective. When one person's genome is corrected, an entire lineage sighs in relief. Generations of suffering end quietly in a single cell dish. With no applause, only gratitude. You feel it radiate through time, a chain of ancestors, whispering thank you across centuries, ethics of care. But even as light fills the horizon, a shadow remains. The power to heal is still the power to change. And change, no matter how pure, carries weight. Scientists pause often, gathering in circles to ask the hardest questions. If we can correct disease, can we also correct desire? Where does therapy end and design begin? CRISPR's promise is golden, but its edge is sharp. Every cut is also a choice, and yet, within those ethical hesitations, there is something deeply human, the acknowledgement that healing must not outpace humility, that progress is not progress if it forgets compassion. In this quiet, you realize the promise of healing is not simply what CRISPR can do. It is how gently we choose to let it, the universe of repair, you drift upward again. Past the lab, the hospital, the earth, the stars return, shimmering like microscopic cells of the cosmos. Somewhere out there, even galaxies break and mend, their spirals torn by gravity, their light reborn in supernovas. Repair, it seems, is not unique to biology. It is a law of existence. The universe edits itself constantly. Cut, realign, recreate. CRISPR is only one reflection of that cosmic impulse. A local dialect of the universal language of renewal. You see it now clearly, the same principle guiding atoms, stars, and cells, the desire to persist through imperfection, healing as harmony. The music of biology begins to play softly again, a low, luminous hum beneath your thoughts. It is the sound of equilibrium, of the body remembering balance. CRISPR does not impose change. It listens, learns, and aligns with what the body already knows, how to heal itself. You imagine your own cells glowing faintly in your veins, each one whispering the same silent mantra: repair, renew, restore. Healing is not only an act of science, it is an act of rhythm, a song the body has always sung. CRISPR simply hands it a clearer note. Transition. The lab lights dim completely now. Only the incubators remain lit, tiny suns of possibility in the quiet dark. You feel peace settle over everything. The machines, the glass, the hands of the scientists. CRISPR's promise is not loud, not grand or heroic. It is slow, molecular, humble, a promise kept one cell at a time. You close your eyes, feeling the same process echo softly within your own mind. Old pain replaced by understanding, old patterns rewritten with care. This is healing, both biological and spiritual. But as sleep begins to fold around you, a question flickers gently at the edge of thought. If we can heal, can we also evolve, not just to fix what was broken, but to grow beyond what was imagined? Night folds softly around the world again. You are no longer in a lab. You stand beneath a vast sky, and above you stretches the unbroken rhythm of stars, each one a reminder that life, too, is born of chaos and order entwined. The earth beneath your feet feels ancient, as though it remembers every mutation, every adaptation, every quiet experiment nature has ever run. It is a breath, the long, patient inhale of the cosmos, learning to become more complex with every exhale. And now, in the hush of modern discovery, CRISPR joins that breath. No longer just a healer, it becomes a collaborator with time itself. The ancient algorithm, long before humans ever looked through microscopes. Life was already editing itself. Each cell, each organism, carried small accidents in its code, mutations that sometimes harmed, sometimes healed, and sometimes gave birth to something entirely new. A fish that learned to crawl, a reptile that grew feathers, a mammal that began to dream. Evolution is the grandest experiment, billions of years of trial and error, written in the quiet ink of DNA. You can almost see it now, a slow dance of molecules, each one trying a new step, each mistake a possibility in disguise. In this endless choreography, CRISPR is a new kind of dancer. It moves with awareness. Where nature once stumbled by accident, we now take careful, deliberate steps, not to replace evolution, but to join its rhythm consciously. The garden imagined you. Find yourself walking through a field at dawn. Dew glistens like starlight scattered across the grass. Each blade hums with life. Each flower carries the quiet pulse of genetic artistry. You begin to see evolution as a garden, wild, ancient, ever-changing. For billions of years, it has tended itself, sunlight selecting for the strong, storms pruning away the unfit, time-rewarding adaptation. And now, CRISPR stands at the garden's edge, a gardener with gentle hands. Learning how to touch without harm, you imagine it kneeling beside a plant, but to help it thrive, to make wheat more resilient, rice more nourishing, plants more patient with drought and heat, in a world trembling under climate change. Such care feels like compassion in molecular form. This is not domination over nature, but conversation with it, a shared effort to survive. Guided change evolution, by its nature, is slow. It does not hurry. It listens, experiments, learns through loss. But humans are impatient creatures. We live in brief spans, always reaching beyond what time allows. CRISPR gives us the means to accelerate that natural rhythm, to guide change within decades instead of millennia. Somewhere, a scientist edits coral genes to help reefs endure rising temperatures. Elsewhere, bacteria are reprogrammed to absorb carbon dioxide, turning the air itself into a cleaner breath. The work is not loud or spectacular. It happens in small flasks, in whispering incubators, in quiet data readouts that could reshape ecosystems. Each edit, each decision, is a question posed to evolution. What if we help you grow differently this time? And evolution, in its infinite patience, seems to nod between ethics and awe. You pause in this imagined garden and feel the tension that hums beneath the soil. To guide evolution is to touch something sacred. To decide what lives and how is to carry the weight of creation itself. You sense both wonder and warning here. For every edit that brings resilience, another might bring imbalance. For every gene made stronger, another species might fade in silence. Science, like evolution, must learn humility. It must remember that control is an illusion, that the garden's true beauty lies in its wildness. Perhaps CRISPR's greatest wisdom is not its precision, but its reminder that precision alone is never enough. We must cultivate not just genomes, but gentleness, the mirror of time. As you walk farther, the garden begins to shift. Plants become patterns, patterns become spirals, spirals become galaxies. You realize you are moving through time itself, watching life's great experiment unfold, from simple RNA strands to cells that learned to remember. Two organisms that dreamed, two humans who could rewrite the code that birthed them. It is humbling. You are both the result and the continuation of evolution, a link in an endless genetic conversation. And now, with CRISPR, the conversation has changed tone. It is no longer only nature speaking to itself. It is you listening, responding, joining the dialogue with care. You are evolution, made aware of its own reflection, echoes in the microcosm inside your body. Evolution continues, even now, not across centuries, but in seconds. Your immune system edits itself each day, learning, adapting, reshaping its defenses. Your microbiome evolves with every meal, every breath, every hour of rest. CRISPR was born from that same microbial intelligence, from bacteria that learned to record viral memories and use them to defend themselves in the future. It's poetic, isn't it? That the tool now reshaping life was itself born of life's instinct to survive. Every edit we make is not a rebellion against evolution, but an echo of it, a continuation of nature's own method, made conscious, made careful. New branches of being you pass a grove of trees, each one shimmering faintly with possibilities. One tree grows fruits that can cure deficiency. Another filters toxins from the air. A third glows faintly, a natural lantern born of genetic fusion between plant and firefly. It is not fantasy. These are real experiments, genes shared between species, boundaries softening in the name of adaptation. It may sound like creation, but it is more like collaboration. CRISPR does not invent new life. It listens to what already exists and rearranges the conversation. You imagine a world where evolution is guided not by dominance, but by empathy, where humans act as stewards, not sculptors, where each change is made with reverence, a gardener's hand, not a conqueror's. The long view you sit now by a quiet stream in the garden. The water mirrors the stars above. They seem closer than before, perhaps because evolution, at its heart, is a cosmic act. From stardust came molecules. From molecules came cells. From cells came consciousness. And from consciousness came tools like CRISPR, which now turn back to look at their origins with awe. It feels circular, a loop of curiosity closing gently on itself. The question is no longer whether we can shape life, but how we choose to do it. Every edit is a ripple in this stream flowing outward through generations unseen. And as you watch the ripples fade, you realize the true art of evolution may not be in the changing, but in knowing when not to. The whisper of the future. The garden fades slowly into mist. You hear faint voices, scientists, poets, philosophers, all wondering what tomorrow will bring. Perhaps CRISPR will help grow coral that survives heat, or crops that restore balance to the soil. Perhaps it will teach us new ethics of creation, or new humility in the face of complexity. Or perhaps it will simply remind us that life itself is an ongoing draft, never finished, always revising. You close your eyes and feel the world breathe. Evolution has always been editing. Now, with CRISPR, you are finally aware of the pen in your hand. And yet, you hesitate, not from fear, but from reverence, because the garden is alive, and you are both its caretaker and its creation. Transition. The air grows quiet again. The stars hum faintly above the horizon. In the soft dark, you feel the next question take shape, like a seed ready to open. If we can guide evolution on Earth, what happens when we look beyond it, toward the stars, toward the fragile possibility of life elsewhere? You exhale slowly, letting that question settle into your dreams. The garden fades, the sky deepens. You open your eyes, and the horizon is gone below you. Earth is a luminous marble, a swirl of white, green, and deep ocean blue, turning silently in the infinite dark. It feels small from here, fragile, like a living cell suspended in the blackness of the cosmos. You float beside it, weightless, and for the first time, you sense what all explorers feel, that life itself is the rarest phenomenon in the universe, and that it must be carried carefully. CRISPR, the tiny molecular editor, is here with you too, not as a tool this time, but as a symbol of how far life has come since it first stirred in the tide pools of an ancient Earth. Origins remembered billions of years ago, before stars had cooled enough to form planets, before oceans ever reflected a sunrise. Atoms swirled in cosmic dust. From that dust came the elements: carbon, hydrogen, oxygen, nitrogen, the alphabet of existence, and from their union, life began to whisper. Not instantly, not miraculously, but slowly, through chemistry's patient art, molecules formed loops. Loops learned to copy themselves. And through endless repetition, a code was born, the same code that now sleeps inside every living thing. You are the descendant of that first whisper. And now, as you drift beyond the planet that raised you, you carry that whisper forward, a story written in the 4 quiet letters of DNA, ATG. See, the softest script in the universe. The traveler's question: what happens when life leaves its cradle? When biology, born of one small blue world, extends its reach toward others? Astronauts and biologists alike have long wondered if we can take life into space, not just to survive, but to adapt, because space is not gentle. It is radiation and vacuum, cold and silence, gravity absent, and time stretched. Yet even here, in this void that seems so empty, life has found its teachers: CRISPR, that molecular memory of survival. Once evolved in microbes defending themselves from cosmic-like invaders' viruses, it remembers danger. It learns. It adapts. And now humanity looks to that same instinct as we prepare to carry life across the stars. CRISPR in orbiting the laboratories aboard the International Space Station. CRISPR is already at work. Tiny vials of bacteria and yeast float gently in microgravity, each cell dividing and changing more slowly than on Earth. Astronauts guide the experiments with gloved hands, their movements deliberate, their breaths echoing through the stillness of the cabin. They are testing how DNA behaves in space, how repair mechanisms falter under radiation, how mutations accumulate in the absence of gravity. CRISPR allows them to edit genes mid-orbit to see how cells respond, to simulate evolution's own experiments in the stars. It is the smallest act of creation in the most infinite of places, a reminder that even here, in the silence beyond atmosphere, life continues to learn, to rewrite, to persist. Seeds of adaptation. You imagine a ship sailing beyond Mars. Inside it, rows of transparent chambers glow softly, each one a miniature Earth. Tiny ecosystems float in sealed bubbles, algae releasing oxygen, yeast fermenting nutrients, plants stretching toward artificial suns. These are the first seeds among the stars, not of trees or flowers, but of survival. Scientists imagine using CRISPR to help these organisms adapt to aliens, worlds, plants edited to resist cosmic radiation, bacteria engineered to digest Martian dust and release oxygen or fuel. Each modification is a note in a new symphony, a song of life retuned for distant harmonies. This is not domination of the cosmos. It is migration, life's oldest instinct. expressed now through molecules instead of wings. Terraforming by breath, you picture a future landscape, Mars. Not red, but green. The horizon still barren, but touched here and there by the shimmer of moss. The ripple of engineered algae thriving in thin air, CRISPR, may one day write the language of such resilience. A code for chlorophyll that tolerates cold. A genetic shield against radiation. Perhaps one day, fields of altered grass will sway beneath alien suns, photosynthesizing a new atmosphere, one quiet breath at a time. It is not conquest, but continuity. The universe reclaiming its own capacity for life. Through us, its conscious children. You smile at the thought, Earth teaching Mars how to breathe, the weight of creation, but even among the stars. Ethics follow. Each genetic change is both a gift and a gamble. What if we seed another planet before we truly understand our own? What if, in our desire to create, we forget to listen to the delicate balance that holds all ecosystems in quiet agreement? The stars do not answer. They only reflect your question back, like a mirror stretched across infinity. You think of the first microbes that altered Earth's atmosphere, cyanobacteria releasing oxygen, turning poison into breath. It was an accident of evolution, but it changed the world forever. Perhaps now, CRISPR is a new form of that accident, a deliberate evolution, one that demands greater mindfulness than any before it. Creation is easy, care is not. The cosmic continuum, you drift farther past the orbit of the moon, past Mars, toward the open sea of interstellar dark. There, the stars hum softly, like neurons in the brain of the universe. In that vast silence, you begin to sense something profound, that evolution was never confined to Earth, that life, once begun, seeks expression everywhere. Perhaps there are other CRISPRs out there, molecular memories written by alien cells, defending themselves from alien viruses, learning their own lessons in survival and change. The laws of biology are not Earth's alone. They are cosmic. DNA is not merely a human script. It is a universal poem, written wherever atoms remember how to listen. And now, through science, you have become one of its authors. A new Genesis, imagine it, a frozen world beneath another sun, waiting quietly for the warmth of possibility. A small lander descends, its metallic legs kissing alien dust. Inside it are vials, the first seeds encoded with CRISPR-crafted life. They are not designed to replicate Earth exactly, but to blend with their new home, to learn its soil, its minerals, its chemistry, and evolve accordingly. A controlled beginning, but an open ending, if they survive. They may one day awaken ecosystems that fit their world perfectly, unique, native, alive in this vision. CRISPR becomes not a weapon of change, but an ambassador of adaptation. Life's quiet handshake with the unknown, echoes of humanity. And what of us, the storytellers, the scientists, the sleepers who dream of other worlds? We, too, are seeds among the stars. Our DNA is a time capsule, not only of biology, but of memory, emotion, and song. Within its spirals lie the stories of ancestors who stared at these same stars from firesides long ago. Now we carry those stories outward, folded into every strand of our being. If someday another intelligence finds traces of our code drifting in the dark, they will not only find genes, but poems, the poetry of persistence, the will to become more than dust, the breath of evolution. You realize now that evolution is not finished. It is only changing scale, from the microbial to the planetary, from oceans to galaxies. CRISPR is one of its tools, a conscious extension of nature's own self-editing instinct. Through it, life learns to write more deliberately, but also, perhaps, more compassionately. You feel a strange peace at this thought. For all the fear and uncertainty of such power, there is also beauty, the beauty of understanding that creation is not random, that even in vast emptiness, life seeks connection. The stars are not cold anymore. They feel like the tips of growing roots, seeking new soil in cosmic dark. Homeward reflection. You turn your gaze back to earth. It gleams below you, delicate, shimmering, impossibly alive. The same blue that gave you birth, the same air that taught your lungs to trust. You understand now that carrying life outward is not about escaping home. It is about remembering it, recreating that fragile balance wherever you go. CRISPR, for all its microscopic elegance, is merely an instrument in that memory, a reminder that adaptation is an act of love, even as humanity dreams of stars. Its truest work will always be the same: to protect life, to nurture it, to help it grow into harmony with its world, whether that world is Earth or something yet unnamed. Transition. You begin to descend, slowly, gently, as if falling back into a dream. Earth's light expands beneath you, and the stars fade into softer hues. The last thought that drifts through you is not of technology, nor of invention, but of continuation. That CRISPR's story, like evolution's, is a promise passed quietly from one generation to the next, a promise not of perfection, but of potential. And, as your body relaxes, as gravity's familiar pull returns, you hear the next whisper, a question forming in the silence. If we can carry life to the stars, can we also learn to carry wisdom, to evolve our hearts as we evolve our genes? The stars have dimmed now. You find yourself once again in the quiet hum of a laboratory, not the shining, sterile kind, but one that feels almost human, a faint smell of coffee, notes scrawled in the margins of notebooks, a soft sigh from the scientist watching her samples glow. The air carries an energy that isn't excitement, but reflection. CRISPR has reached a point where discovery and responsibility meet, and the space between them is thin as light on glass. Tonight, the work is no longer about what can be edited. It's about what should be. The moment of power. There's a story in every lab, a moment when a researcher realizes the weight of their experiment. It's rarely cinematic, no thunderclap, no flash of revelation. It's quiet, arriving between the click of a pipette and the hum of a centrifuge. You feel that moment now. Imagine editing an embryo, correcting a genetic disease before it ever causes suffering. A beautiful thought, isn't it? But imagine, too, that the same technique could alter the color of eyes, the height of a child, the shape of intelligence. The line between healing and design begins to blur. And once blurred, it's so hard to see again. CRISPR gives humans a power evolution never reserved for one species alone, the power to rewrite the script of life consciously, And with that comes the oldest question whispered by creation itself. Just because we can, should we? Echoes of the past science remembers its shadows. History keeps them close. You recall the eugenics movements of the early 20th century, where people tried to define better humans through ideology instead of empathy. It ended not in progress, but in horror. Now, CRISPR stands at a similar crossroads, but with much quieter tools. This time, the edits are not made by politics, but by precision. Yet the moral weight remains the same. Every cut in DNA is still a cut in possibility, the trimming away of one future to make space for another. And the ethics of creation lie in choosing which futures are worthy of growing. You sit with that thought. It is heavy, but it is necessary. The quiet voices ethics is not only written in policy documents or declarations, it is whispered in the hearts of those who work late into the night, staring at cells glowing beneath the microscope. Scientists to dream. They feel the ache of parents who lose children to genetic illness, the longing to heal what evolution left unfinished, and yet they also carry fear that healing without humility can turn into hubris. Some say CRISPR should never be used on embryos, that altering germline DNA, the kind passed to future generations, crosses a sacred threshold. Others argue that not using it when we could prevent suffering is its own kind of cruelty. There is no perfect answer, only balance, only awareness that every discovery demands reflection equal to its power. The line between hope and hubris, the stillness. You imagine a future laboratory. A parent stands beside a screen, watching their unborn child's genome unfold like a constellation of letters. A scientist points gently. This sequence may cause disease. We can fix it. The parent hesitates. Can you also change their eyes, their memory, their kindness? The silence between those questions feels infinite. In that silence lives the essence of ethics, the understanding that science can map biology, but not morality. The genome is not a canvas for preference. It is a tapestry of ancestry, diversity, unpredictability, the very chaos that makes love and life meaningful. If we edit too perfectly, we risk erasing the imperfections that gave us empathy in the first place. The story of Lulu and Nana. In 2018, two twin girls were born in China. Their names, given publicly, were Lulu and Nana. They were the first humans whose DNA had been edited using CRISPR before birth. The scientist behind the experiment claimed to make them resistant to HIV. A medical goal with moral complexity, but the world was not ready. The experiment broke ethical guidelines, ignored global consensus, and introduced unknown changes into their genomes. The reaction was immediate: outrage, fear, disbelief, because science is not just about success, but about consent, transparency, and shared understanding. Lulu and Nana became living reminders that power without patience can lead not to progress, but to pause, to silence, to rethinking what it truly means to create responsibly. The ethics of care. Imagine a scientist placing a hand on a cultured dish, not to manipulate, but to remember the fragility of what lies within. Ethics begins with care, with the quiet promise that creation must serve compassion first. Every experiment, every cell line, should be guided by a question deeper than data. Will this bring balance to life or burden? CRISPRs, scissors, can repair genes, but ethics are the steady hand that decides when to lift them. True progress is not measured by speed or precision, but by wisdom, the wisdom to stop, to ask, to listen to the quiet before cutting the thread. Because every sequence of DNA represents not just chemistry, but ancestry, memory, the continuity of living history, the circle of consent creation, if done ethically, is never an act of isolation. It is a conversation. Parents, patients, scientists, philosophers, each voice matters in shaping how we move forward. In some places, global councils now discuss the boundaries of human gene editing. In others, indigenous communities remind the world that life is not a resource to be owned, but a relationship to be respected. The ethics of CRISPR are not just scientific. They are cultural, spiritual, emotional. Every society will answer differently, but the act of asking together is what makes the process ethical. Ethics, like evolution, is alive. It adapts, grows, learns, and so must we. Nature's own reminder, outside the lab, the night stretches endlessly. The stars above you shimmer like living cells, each one born from explosions, yet balanced by gravity's gentle pull. Nature itself is a system of checks and balances. Every act of creation is countered by the laws of equilibrium. Too much fire, and a star collapses. Too much restraint, and it never shines. Ethics is our human gravity, the unseen force that keeps creation from imploding. CRISPR, for all its elegance, needs that gravity now more than ever. Because we are not separate from nature's patterns. We are their conscious expression, and consciousness demands care, humility in the code you think of DNA again. The 4 simple letters repeating endlessly, A, T, G, C. In their simplicity lies humility. They do not boast, they do not choose. They simply exist, allowing life to unfold as it will. Perhaps our ethics should mirror that rhythm, humble, observant, patient. CRISPR allows us to speak the language of life. But like all languages, it requires listening before speaking. Creation without humility is noise. Creation with humility is music. And ethics is the pause between notes that makes the melody meaningful, the mirror of responsibility. You look into a glass pane separating you from the experiment. In it, your reflection shimmers faintly, superimposed over the glowing culture dish. It is a haunting image, because in that reflection, you see both the creator and the creation. Humans are editing the code of life, but that code has, in turn, written humans. We are both author and text, reader and story. To change one is to alter the other. Responsibility, then, is not a burden, it is an acknowledgement of kinship between scientist and subject, between life and the laws that sustain it. To edit without reverence is to forget we are editing ourselves. The sacred pause. Imagine a moment before every CRISPR cut, a breath, a heartbeat, a stillness where intention is examined. That pause is sacred. It separates invention from impulse. Ethics is not about restriction. It is about rhythm, knowing when to move forward and when to wait. Even stars rest before they collapse into new ones. Even evolution pauses between epochs in that stillness. Creation listens for permission. And perhaps that's what ethics truly is. The universe asking itself, quietly, is this act aligned with care? The light of awareness morning begins to seep into the laboratory windows. Soft gold spills across the counters, turning instruments into silhouettes of promise. You feel something shift, not in science, but in understanding. CRISPR is neither good nor bad. It is a mirror reflecting the intentions of those who wield it. If guided by compassion, it heals. If guided by ego, it harms. The difference lies not in technology, but in awareness. And awareness is the most human of all genes, the one that has allowed us to dream, to doubt, to ask what is right, even when no one is watching. Transition, the sun has risen now, turning the lab into a cathedral of light. Petri dishes glint like stained glass. The hum of machines sounds like prayer. You realize that ethics is not a boundary, but a compass, a quiet inner voice reminding creation where home is. And as you prepare to leave, you glance once more at the incubator, where edited cells divide in calm, rhythmic grace. They are not just data points. They are possibilities, and the future depends not only on what we make, but on how gently we choose to make it. You close your eyes. The light through your eyelids feels like warmth and forgiveness. In that warmth, a new thought rises, a final step in this long journey. If ethics guides creation, what guides humanity itself when our tools become wiser than we are? That question shimmers softly, leading you forward. The night has given way to a pale gold morning. Outside, the air feels newly washed, trees dripping with dew, birds tuning their first hesitant notes. The world feels delicate, like it's been rewritten in the smallest ways. Without you even noticing, you breathe, and in that breath, countless molecules move through you, oxygen shaped by plants, carbon exhaled by ancient volcanoes, atoms forged in the hearts of stars. You are, quite literally, the future built from the past. The revolution that began with CRISPR is not merely a story of tools or technology. It is a continuation of life's oldest impulse to adapt, to evolve, to survive through creativity. And as the light deepens, you realize something almost spiritual. The future of biology is not about changing what we are. It's about understanding, with reverence, what we already contain. The living library. Every cell in your body holds a complete copy of your genome, a library of instructions written in 4 letters, A, T, G, and C, 30 trillion cells, each carrying the same silent script, each reading it differently depending on its role in the grand organism that is you. A neuron in your brain reads the pages on thought, memory, and emotion. A skin cell reads the chapter on protection, while a liver cell studies detox and transformation. Like a choir singing from one score, your body performs the same composition, each voice distinct but harmonious. CRISPR, that elegant molecular tool, is simply a new way of editing the notes, fine-tuning a melody that nature has been composing for billions of years. But what does it mean to change a line in a book that's been passed down since the dawn of life? It means carrying a responsibility not just to yourself, but to every living story that came before you. Life's continuum. You imagine life as a great river. At its source are the earliest cells, simple and unknowing, dividing and adapting in the oceans of the ancient earth. Downstream flow countless generations, plants, fish, mammals, and finally humans. each ripple carrying echoes of the one before. CRISPR is like a hand reaching into that current, not to stop it, but to guide its direction slightly, to clear a blockage, to deepen the flow, to let life continue more freely. Yet even the gentlest touch changes the pattern of the river. The ethical weight, the humility, must always accompany the hand. The river does not belong to any one traveler. It belongs to time. And you are both the traveler and the water itself, the future's quiet shape-in labs across the world. Scientists continue to explore what CRISPR can become. There are trials that repair blindness, that restore red blood cells to healthy form, that silence genes causing cruel diseases. Each success feels like a lantern lit in a darkened corridor, a glow of hope carried forward. Yet, what makes this future remarkable is not the technology alone, but the intention behind it, the promise to use knowledge, not to dominate, but to nurture. Imagine a time when medicine becomes truly personal, when your own cells are your healers. When a doctor listens not only to your heartbeat, but to your genome's quiet needs. Imagine a future where no one inherits unnecessary suffering, where prevention replaces correction, where evolution becomes a partnership between biology and consciousness. This is the future. CRISPR whispers toward, not loud, not abrupt, but gentle and wise, guided by care. The symphony of collaboration science no longer moves in isolation. The great discoveries of this century are collaborations not just between scientists, but between disciplines, between countries, between ideas. CRISPR itself was discovered because a handful of microbiologists listened to what bacteria were saying through their DNA. It took linguists of life to realize that these microorganisms had invented a natural system of memory and defense, and it took molecular biologists to translate that microbial wisdom into a human tool for healing. The future of biology will continue this pattern. Scientists listening to nature, interpreting, not commanding, because nature, when heard closely, already knows the answers. You realize that perhaps the next revolution won't be about control at all, but about listening deeply enough to understand how life wishes to grow. The code and the consciousness. Close your eyes for a moment. Within you, neurons flicker like galaxies, each one firing tiny sparks of electrochemical thought. They, too, follow biological patterns, synapses, strengthened by repetition, weakened by neglect, yet consciousness arises from them, something no gene can entirely explain. If DNA is the hardware of life, then consciousness is the soft light that animates it, and here lies a mystery. We can edit genes, but can we ever edit awareness? What makes kindness? What encodes wonder? What part of you decides to look at the stars and feel small, yet infinite at the same time? CRISPR may change our biology, but consciousness changes what biology means. It transforms a molecule into a memory, a cell into a soul. The future of life will depend not only on how we design our genes, but on how deeply we understand our own minds. The garden within you. Imagine your body as a garden, 30 trillion living things growing, dividing, repairing, a rainforest of cooperation in your gut. Microbes whisper chemical conversations. In your blood, Enzymes carry oxygen like couriers of flame. In your DNA, ancient viruses sleep, remnants of battles won millions of years ago. CRISPR works here, too, quietly cutting out harmful sequences, awakening helpful ones. But what if, beyond editing, we learn to tend the garden instead, to feed it with balance, to let diversity bloom, to guide without domination? Because the secret of every garden is not control, it's care. And perhaps that's the softest truth of all. The most powerful future will not be engineered by force, but cultivated through love. Children of stardust, encode them. More you think about genes, the more you realize how ancient they are. The atoms in your body were once inside collapsing stars. The carbon in your skin once floated in clouds of interstellar dust. And the DNA within you is a living archive of every organism that has ever survived. Life is not new. It is eternal, passing from one form to another. Learning, adapting, remembering, CRISPR, is simply the next way life remembers itself, a bridge between chemistry and consciousness. When we edit a gene, we're not changing something foreign. We're conversing with our ancestors, continuing their endless experiment in existence. You feel it now, a quiet unity, as if all time is folded within your cells. You are not living in the future. You are the future, breathing. The moral genome beyond, the double helix of DNA. There may exist another strand, an invisible one, woven of empathy, awareness, and ethics. Call it the moral genome. It evolves not through mutation, but through understanding. Each generation refines it, makes choices that ripple forward like waves. CRISPR gives us the ability to change the biological script, but it also challenges us to refine this moral genome, to edit cruelty into compassion, ignorance into curiosity, fear into responsibility. Because if we are to redesign life, we must first redesign the way we see it, not as raw material, but as sacred narrative. The next revolution, the quietest one, will happen not in labs, but in hearts. Echoes of tomorrows. You imagine Earth decades from now. Perhaps diseases that once devastated humanity will be reduced to memories. Perhaps extinct species will return, their DNA revived from preserved fragments. Perhaps crops will grow in barren lands, nourishing billions. And perhaps we will finally learn that the greatest technology is not invention, but intention. Every CRISPR edit, every medical breakthrough, every spark of human thought will carry a question. Does this act honor life? If we can hold that question gently, without arrogance or fear, then the future will unfold like the dawn you're witnessing now, slow, soft, and full of promise. The breath of continuity you breathe again. Air moves in and out, invisible, simple, vital. Each breath connects you to every living thing that ever breathed before you. Dinosaurs, mosses, whales. The first bacteria on a volcanic shore, they all shared this same motion. Inhale, exhale. Life continues, and so, too, will the story of CRISPR, not as a single event, but as a continuum of creation. Each discovery guided by the gentle rhythm of care. The future is not built in one moment of brilliance. It is woven breath by breath, choice by choice, like the double helix itself. The still pointless twilight returns. You sit in quiet reflection. The world outside hums with its usual sounds, wind through leaves, the distant call of life continuing, you realize that the future isn't coming. It's already here, flowing through every pulse, every thought, every cell dividing quietly inside you. You don't need to chase it. You only need to listen, because the greatest revolution is not in changing what life is, but in understanding what life means. And tonight, as you drift toward sleep, you carry that awareness with you. The awareness that you are both the observer and the observed, the editor and the edited, the creation and the creator. The heartbeat slows. CRISPR, the quiet revolution, continues its work in the background of the world. But here, in stillness, the future feels less like technology and more like tenderness. You are part of it, a cell among stars, a thought among generations, a fragment of life forever rewriting itself toward love. The story rests now, not with an ending, but with an open pulse, a soft glow, because the future has always been within you.